is Shushmita. So the topic that I am going to discuss today is histidine operon. Okay. So before getting into histidine operon, first let us discuss what is operon. Operon is basically a group of closely linked genes, which include structural and regulatory genes. So basically, these genes are involved in the regulation of a metabolic process. So these are mainly found in prokaryotes and not in eukaryotes. Um, and the thing is, these genes can be in off mode or on mode. Based on that, the operon will be functional and non-functional. This mechanism is being explained as operon model usually. So what is histidine operon? So as I told, with the basic definition of operon, we must already know what is histidine operon. So basically, it's an operon which is involved in the synthesis of histidine. So histidine is encoded by 9 tightly clustered genes in a single operon. That is single operon, and that operon is large operon. So this operon is also called as his operon because it's histidine operon. So okay, so the biosynthesis of histidine is controlled by two methods. One is by inhibition by histidine. So basically, what happens is when histidine enters into the cell, the intracellular concentration is increased, and because of this, feedback inhibition takes place. Feedback inhibition is something when, for example, if the the substrate which has to bind is more in concentration, the substrate itself will inhibit the process, inhibit the synthesis of, or inhibit the reaction over there. So substrate over here is histidine. So basically, if histidine is more, it's going to inhibit its own action. That is one method. And another method is repression control. Repression control is basically that where the enzymes control the action or reaction of the operon. Okay, so let's get into the structure of operon. It's operon. So histidine operon has 9 genes, as I told earlier. Those are G, D, C, V, H, A, F, I, E. Uh, the picture over here is not that clear. I don't know. I don't really know why. But yeah, these are the genes. Each of these box are each genes, that is G, D, H, and so on. These basically encode for enzymes which are involved in the pathway. These enzymes are uh, basically uh, which are involved in the synthesis of histidine from 5-PRPT, that is 5-phosphoribosylate 1 pyrophosphate. As I told before, this is a polycystonic mRNA, operon, which has around 7,300 nucleotides in them. And it's the promoter is row dependent. Sorry, the promoter is primarily present, and then the terminator is row dependent. Okay, so the control. Okay, so basically, this slides explains the transcription initiation and elongation of the histidine operon. So histidine operon is generally regulated by the attenuation of transcription. What that basically means is that there are histidyl tRNAs present. Based on the level of the concentration of these tRNAs, the histidine biosynthesis happens. The enzymes of these are histidine biosynthetic enzymes are being uh, synthesized. So yeah, there is a 16 residue coding sequence which codes for continuous 7 histidine molecules, which is like a marker for the transcription to initiate. Only after there is 7 molecules of histidine present by the 16 residue coding sequence, that is when the initiation will start, and then the termination is row dependent. I will explain that in the next slide. So basically what happens is, as I told, there is a 16 residue strand over here, which synthesis a continuous sequence of 7 molecules of histidine, these molecules. Once these molecules is being synthesized, they form loops. The other strands over here has a lot of G content. So basically they form stem and loop structures over here. And once they form this, after that it is the poly U sequence, which basically terminates the transcription over here. This method is called attenuation, and what happens over here is the ribosome will come until this sequence, until the B region, right after the 7 molecules of histidine is being synthesized. That region is called B region. Till there, if the ribosome comes and synthesize the uh, histidine, then it forms a loop at C and D, and then again a loop at E and F, which causes the poly U to occur, which terminates the entire process. This is called attenuation. But in case of normal transcription, what happens is the B and C region gets looped up, and then D and E region gets looped up. So basically, the F region is still left for the transcription to happen, and then the poly U sequence is not yet formed. So the transcription just gets on going on, or it just elongates, and it doesn't terminate. Okay, so let's get on the negative regulation of the operon. So as I told before, the operon has 16 residue sequence, which synthesize the A region, that is the 7 histidine molecules. So basically it's the same process what I told in attenuation that happens in negative regulation. So what happens here is the ribosome is very much free to move, because yeah because tRNA, the histidine tRNA is more in number. So what happens here is, when histidine is more in number, automatically the histidyl tRNA will also be more in number. So as the tRNA is more in number, the transcription also happens continuously. So the tRNAs keep binding to the ribosome, and the ribosome keeps shifting as soon as possible, and then moves forward. So that is why it moves till the B region, and then again forms a loop at C and D and E and F. So basically again the poly U tail will come, and then there are no synthesis of structural genes, so there is no synthesis of histidine. Therefore, it is to observe that when there is high number of histidine molecules already present, then there is no synthesis of structural genes for it, which is basically what I told in the beginning. The histidine itself is going to inhibit its own process here in negative regulation. Let's get on to the positive regulation. So what happens in positive regulation is, when histidine level is already low, the histidyl tRNAs also are very low in number. So the ribosome doesn't get a chance to move forward in very much fast motion. So that is why the ribosome only moves till the A region. Uh, After A region, it just moves very slowly. That is why the loops are formed between B and C and D and E. and again the F is left out. This is exactly what I told in the attenuation step over there. Just that I didn't tell you which is negative and which is positive there, because I wanted to relate over here with low and high levels of histidine. So yeah, again, because of this F region, the structural genes are being synthesized, and these structural genes will help in the synthesis of enzymes that are involved in histidine biosynthesis. So that's how histidine is synthesized. This is basically the conclusion of whatever I have told till now. This is the loop over here. And then when histidine is low, they form loops, and then there is no place for them to form structural genes. It just ends. And then, yeah, sorry, start around. Reference. These are my references.